I just wanted to give a quick little intro, and then I'm going to give the time to Bailey, who is the president of the Evolution of Bioinformatics Club, to introduce our speaker for today. But I hope everyone's been taking advantage of the things that we have out there. If not, after, go back out. There's a bunch of stuff that's free. There's T-shirts to buy. Don't forget to get a T-shirt, $15. And right after this, we're going to have birthday cake. So as soon as we're done eating here, we'll all walk out there, sing happy birthday, and we'll eat cake together. All right. And then I wanted to also announce that I had Jessica put this up on the very beginning of her slide deck, but she's speaking a second talk tonight. It's not the same thing she's doing here. It's two different talks. And that's going to be at the Life Science Bean Museum up at BYU. And there's a reception after that. And it's also a good opportunity to go and see the brand new Tree of Life exhibit that they have up there in that museum, which is fantastic. So I highly, if you can go, this is going to be a great opportunity to once again listen to Jessica, to also interact with her at the reception, and then see the Tree of Life exhibit. Okay, so I think those are the main. Oh, I wanted to thank Carl Gelman. Is Jess here? I don't see Jess, but Carl Gelman is my co-organizer from faculty. Jess Cusick also helps from the faculty. There's other faculty that also help. I want to thank everyone who brought stuff for booths. Melissa from Zoology, Botany Club, what's it called? BioHive Club, and Evolution Bioinformatics Club. Kyrie brought her wonderful butterflies there. So a bunch of us came together to try to do something cool. So hope you're enjoying Darwin Day, and I'm going to turn the time over now to Bailey.
Hi, everyone. Thanks for coming. I'm Bailey Genesey. I'm the president of the Evolution Bioinformatics Club. And then today, for Darwin Day every year, we invite a speaker. And this year we have Dr. Jessica Ware. Just as a little introduction, she is an evolutionary biologist serving as the curator and division chair of the invertebrate zoology at the Museum of Natural History. She does a lot of work in dragonflies and damselflies. And beyond her research, she also does a lot of leadership at the sites of the community, having served as president of the Entomological Society of America, the Society of Systemic Biologists, and the Worldwide Dragonfly Association. She is also deeply committed to science communication and mentorship, sharing her work through with audiences through public outreach and media, including the documentary we had played out there all day today. That one was called Bugs That Rule the World. And we're so excited to have you. We do have a gift.
Thank you so much. Halloween pendant.
There we go. This is Dr. Ogden actually really does a lot of watercoloring. So we drew watercoloring and then it's a t-shirt.
Yay, can I get a picture?
Thank you so much.
Someone take it with me, please?
Awesome. This is beautiful. So shiny. I put it here as a prop to remind myself what I'm talking about. Oh no, it's dragon. Okay, perfect.
And then I'm going to pass the time over to Dr. Ware.
Thank you so much. Well, thank you so much
for the mic on.
Oh, yeah. Can you hear me? Mic should be on. That's fine.
Oh. All right, one more time, flip it.
There we go. Perfect. Thanks, Bailey, for that lovely introduction, and thank you so much, Keith Ogden, for the invitation to come here and talk to you about my favorite group of animals, which are dragonflies and damselflies. So I feel very lucky to be here talking to you on Darwin's birthday. But I also feel like it's kind of remarkable because I was not raised by people who did science or who were interested in evolution at all. I'm one of a set of twins, and no one in my family is a scientist. So my mom was a high school art teacher before she retired. She also does watercolors. It's one of her paintings up there. And my dad, she's from Canada. We were raised in Canada. My dad was a Marine Corps drill instructor who later went on to do computer sales at Digital Equipment Corporation. So definitely nobody in my family really were encouraging me to read about Charles Darwin or really encouraging me to learn about evolution. But thankfully, I spent a lot of my early childhood with my maternal grandparents, Harold and Gwen Irons. They lived in Northern Ontario. My granddad finished grade 8, I think. My nan went on to be a homemaker. They really had no formal kind of schooling or education in science, but they loved plants. They loved gardening. They liked hiking. When they would take us on these hikes, they would flip over rocks, hand us a snake, hand us a frog, and they encouraged my twin and I to ask questions. Why are there so many colors of dragonflies? Why do you think it is that that frog is only found in this one part of the forest that you go to? And that kind of curiosity was a gift. And that curiosity is what led me to become an evolutionary biologist. But it wasn't like I just started out as a kid thinking, now I'm going to be a scientist. I just knew I liked doing things outside. I mentioned to the students in the break earlier, I also didn't like being told what to do, kind of stubborn kids. And I really liked swimming. And we spent a lot of time swimming in lakes in Northern Ontario. So one of my parents' friends said, if you really like all of those things, I think you should be an oceanographer. So I just memorized that word. I'm going to be an oceanographer. And I moved thousands of kilometers from Ontario to Vancouver, to the University of British Columbia, where I signed up for oceanography. And lo and behold, my first week of classes, I realized, I don't want to be an oceanographer. Oceanography is physics, the chemistry of waves. And my TA said, girl, I think what you want is marine biology. You're interested in the animals that are living in these waters. So I quickly switched to marine biology, and I was taking a lot of invertebrate zoology classes. And Karen Needham taught the invertebrate zoology class section on entomology. And she came in and she said, there are more insects in terms of abundance and species number than there is anything else on Earth. And that kind of got me hooked. And I, from that moment on, worked with a variety of mentors who first told me that you could get a job as a professor. They first told me what tenure was. They first told me about grad school and encouraged me to apply. And if not for them, I really wouldn't have done any of those things, and I wouldn't be standing here now. So really, I'm here because I'm a product of mentors who took the time to talk to me and to teach me about things that they cared about. I still thought maybe I was going to do something very practical. All of the work I did at UBC as an undergraduate assistant was about biological control and integrated pest management, protecting food security, saving human health. Those are lofty goals. But when I went to Rutgers for my PhD, I started working on dragonflies and also on Dictyoptera, and my advisors were evolutionary biologists. And once you learn about evolution and you learn all the wacky things that have happened over hundreds of millions of years, I mean, how could you not become a systematist? Especially when you're talking about dragonflies and damselflies. There's such diversity, shape, form, size, color, behavior. There's really a lot of questions that you could ask. I was talking to the students earlier and mentioned that in the beginning, I felt a little bit insecure. Surely dragonflies are so beautiful that everything would already be known about them. How could something that's so charismatic and colorful and amazing, how could there still be questions to pose? Well, it's been 20 years, a little more than 20 years ago, 25 years, and we're still asking a lot of questions. The group that I first started working on was a superfamily called the Libelluloidea. There was a little over 1,600 species in this group, and I'll talk to you a little bit more about, because we still work on that now. But it was while I was doing my PhD that I also started working on these things. Beautiful, right? We can all agree these are beautiful. Everybody agrees?
Yes, of course.
They're beautiful. Cockroaches. And this was the first time when I was in graduate school that I learned about the thousands of species of cockroaches that are not pests. Only 2% of cockroaches are pests, and the rest of them live kind of outside of the human condition. I was really interested in one particular type of cockroaches. Those are the social cockroaches, or the Isoptera. At the time when we were working on this, you might be surprised to know, we really weren't sure how mantises, cockroaches, and termites were related to each other. It turns out that termites are just fancy social cockroaches that do kind of this interesting behavior. So now for my job, I'm a curator at the American Museum of Natural History. And as a researcher there, I'm responsible for a lot of different types of collections. But importantly, we have the world's largest termite collection. So 80% of the world's types are at the American Museum of Natural History. So this is a pretty great job, I would say. I'm really glad that my mentors encouraged me to do this, because as a field biologist, as an evolutionary biologist, I travel the world so that I could look for specific individual taxa that I need so that I can understand their evolutionary history. Or sometimes we go to a region because we want to understand the biodiversity in a particular area. And whenever we go to do that, I'm always amazed at how much diversity is out there. Karen Needham was right. There are a lot of insect taxa out there. I do a lot of my work in South America in a coastal nation called Guyana. These are some photos of us when we were last there earlier in 2025. There's a lot of former Utahns who have gone with me. This is former BYUer Ethan Foman and Ellie Gamet. And in doing all of this work, what surprises me is just this amazing biodiversity. So my children, I have two kids, a 21-year-old and a 17-year-old. They learned a lot in school about mammals. And I learned a lot in school about mammals. There's only 6,500 mammals. There's 1.5 million described species of insects. That blows my mind. And some people say there's more. Some people say maybe it's 10 million. Some people say it's more than 10 million, with the majority of taxa yet to be described, yet to be discovered, never mind described. So we can use phylogenies, we can use our understanding of evolutionary history of insects to ask questions about when certain taxa diverged, where they diverged, and maybe what drove some of that diversity. So a lot of what we do is we make phylogenies or trees where we have the taxa that we're interested in and we connect them by lines. In this example, the camel and the bird are kind of connected and they form a clade, which means that they share a common ancestor. They're more closely related to each other than they are with the fish that's in this example. We can do that with a lot of data. This is a ginormous tree that has over 1,000 species in it. This is using transcriptome. You make phylogenies with molecular data. Technically, I mean, you can make phylogenies of everything. There's a phylogeny of sandwiches. Like, where does a taco fit in the phylogeny of sandwiches? People have it. You can Google it. You can make phylogenies of everything. But we can make phylogenies with molecular data and try and understand how insects are related to each other. And when we do that, at the very base of the tree, the earliest branching lineages is the Paleoptera. And this is what Heath and I have spent our lives working on: the mayflies, the dragonflies, and the damselflies. So for the dragonflies and damselflies, they are the order Odonata. And what a group, man. Probably first to fly, although we could debate about that, whether it's mayflies or dragonflies and damselflies. Probably the first to fly, before birds, before bats, before pterosaurs. Nothing in the sky, something that looked maybe like a dragonfly. The juveniles develop in freshwater. Females lay her eggs in freshwater, and they're voracious predators both in the juvenile stage and in the adult stage. They vary a lot in size, from tiny things like Nanothemis bella to the giant pseudostigmatic damselflies that you see in Central and South America. Some dragonflies and damselflies fly 11 meters in their entire life. Sometimes they never leave the pond from which they emerge, and others migrate around the entire globe. They're able to do this because they have remarkable flight, and a lot of that has to do with their wings. They also have remarkable vision. And part of that vision is because they need to be able to communicate with each other, and colors are a really big part of their story. And then lastly, I'll just mention, they do have interesting reproductive behavior. So males have 2 sets of genitals, and they transfer sperm with a method called indirect sperm transfer. So all of these are good reasons to really dive into a group that has the same number of species as all of mammals. There's around the same number of species of dragonflies and damselflies as there are all of mammals. So we basically we like to group things as humans, right? This is kind of what Darwin was doing. He was kind of categorizing and grouping things. That's what we've done for odonates. We group them into 3 main groups. The dragonflies, which tend to have very stocky bodies, and they hold their wings out to the side when they're at rest. Then there are the damselflies, which have very slender bodies, and they tend to hold their wings behind their back when they're at rest. And then there's this third suborder. It doesn't have a common name. It's called the Nizozygospora. It has a stocky body like a dragonfly and thin petiolate wings like a damselfly. You can only find this in China, the Himalayas, and Japan. This is what the juveniles look like. They have a labial mass that kind of shoots outward, grabs their prey, and brings it back into their mandibles where they eat. Dragonflies have their gills so that they can breathe underwater inside their bum. Damselflies have external gills called caudal lamellae. And what they're eating, as I said, they're really voracious predators. They eat each other. They eat other insect larvae, but they can also take down vertebrates. So they can eat tadpoles and they can eat small minnows. So they've been doing this, we think, for a very long time. The fossil record suggests that we have fossils of modern dragonflies for at least 200 to 250 million years. But we also have these things called griffin flies, which flew during the Carboniferous period. Each of these ancient lineages, they had wings that were about 37 centimeters. So it's about a 2-foot wingspan. These griffin flies, we think, were probably pretty poor flyers, kind of clumsily flying with very stiff wings as they paddled through very viscous, high oxygen air. And they also had a beak, just to kind of add an extra component to their ferocity. Yarmila Kookaburra Peck did a lot of work on this group, and she suggested that they also had juveniles that were in fresh water. And they also, the juveniles, the fossils that she found, had labial masks that possibly shot up just the way that modern dragonflies have. So although we have these griffin flies, those are not what we call crown or true odonates. True odonates are, of course, much younger. So we can make phylogenies, we can use different types of data to ask questions about how dragonflies and damselflies are related to each other. And we've done this. This is a phylogeny that Seth Bivey and some colleagues and I did using around 500 genes. We've done this with the insect transcriptome group using around 3,000 genes. And basically, we're starting to converge on the same answer. We think that damselflies evolved first, then that third suborder that has body like a dragonfly, wings like a damselfly, that is sister to the rest of the group, which are the dragonflies. We have this lofty goal. Mammals have done this. We have sequences for almost all the mammals. What if we did that? We have about the same number of species as mammals. So we have this lofty goal to generate molecular data for all of the Odonata. And we're about halfway there. And a lot of this has been driven by our former postdoc, Lacey Newton. I'll just tell you a brief highlight of what we found out about how these animals are related to each other. Of course, we use mayflies as the outgroup, as a sister to the rest of the dragonflies. And when I was first starting to work on dragonflies and damselflies, there was this hypothesis that maybe there was this one tiny damselfly that was sister to the rest of all of the odonates. It's called Hemiphlebia mirabilis. It looks like this. It has kind of a green metallic body. And the females do something really weird. Instead of laying her eggs in plant material in the water or on the surface of the water, she actually kind of arches her body, her abdomen over her back, and puts her egg there. And then she just chucks it. And she kind of just chucks her egg somewhere in the wetland. Is this a good strategy? I don't know, but I will say that they are extremely endangered. And it is today going to fragmenting the population. So that's where they might go in our lifetime. But the question was whether or not that was going to be recovered with our molecular data as sister to the rest of the odonates. And it's not, but it is one of the earliest branching lineages within the damselflies. A lot of stuff is happening in the damselflies, but I'll just highlight a couple of my favorite groups of damselflies. These are the Polythoridae. These are found in the Western Hemisphere. But what's so cool about them is they have a lot of cool color. Maybe they're part of a mimicry ring with toxic butterflies, Theomine butterflies. But look at the nymphs. They've modified their external gills to do a function. So these damselflies, their babies develop in waterfalls. And waterfalls, that's a lot of fast-flowing water. So they actually have modified their gills to no longer function for oxygen exchange. And instead, they're like maces, kind of. They're like claws. And they use their gills to cling to the rock face as the waterfall kind of flows over them. But they still need to be able to breathe. And they actually have lateral gills. They have lateral gills along their abdomen, which is unusual because we think of mayflies, that's kind of their story, having a lot of lateral gills. But dragonflies and damselflies in general do not have lateral gills, except for Polythoridae. And this other group called Euphaidae. Euphaidae have done something different with their caudal lamellae, with their external gills. They've modified them almost to be like a little gaseous sac. And when you see them, they're in kind of very shallow streams, and their gaseous sacs kind of hold their bum right at the surface of the water. And they also have lateral gills, like finger-like projections that kind of extend along their abdomen. There's around 3,000 species of damselflies. I don't really have time to tell you all the stories about them. Let's jump to the group that's my favorite, which are the dragonflies. So that third suborder, the one that doesn't have a common name, kind of Himalayas in Japan, body like a dragonfly, wings like a damselfly, you might wonder, what do his gills look like? Are they internal or are they external? They're internal. So they actually look an awful lot like a dragonfly, which makes sense because that's a sister to the rest of the dragonflies. Then with the dragonflies, one of the first branching groups are these really fast flying, colorful dragonflies, the Aeschynidae and the Australopithecinae. These can fly like 30 miles an hour. A lot of them are blue, they're not all blue. Aeschyna canadensis is my most favorite dragonfly. I love it so much that that's the name I actually gave to my daughter. Her name is Aeschyna. And this is what the Aeschyna dragonfly nymphs looks like. Another group that we've talked a lot about in odonatology, trying to understand their evolutionary history, are the petal tails and the Gomphidae. And historically, people really couldn't tell how these two groups were related to each other. One has only 11 species and one has 1,000. So why is one so species poor and one so species rich? And then add on top of that, there's this Sasquatch connection, because there's a fellow named Perry Turner who in the 1970s was at Berkeley, and he was studying the petal tails. He spent a lot of time in the Pacific Northwest studying Cynipteryx. And while he was studying Cynipteryx, he became convinced that Cynipteryx nymphs were the primary diet of Sasquatch. And Sasquatch are rare because Petaluridae are actually very rare in the habitat. Nymphs live in kind of spring seeps and in fens. And he went further to say that if you were to eat the nymph of a petal tail, you would become a Sasquatch. Now, he didn't just say this, he typed it into his PhD thesis and he got his PhD. So for the students in the audience who are thinking, am I good enough? Well, do you have Sasquatch data in your PhD? Probably not. You're probably doing great. You can see this thesis. It's at the Smithsonian. It's not at the Smithsonian, but the Smithsonian just happens to have all of the contents of his storage locker because he disappeared in the 1990s. Some say he went to live with a Sasquatch, and a lot of his really beautiful biological illustrations and his thesis are now at the entomological department. So we often think of these as being Sasquatch food. So my graduate student, former BYUer Ethan Tolman, he was really interested in this group. He still is. And what he found is that this very, species poor group seems to have had a Laurasian clade, a northern hemisphere clade, and a southern clade that evolved during Gondwana. And what's interesting about this Sasquatch food group is that each individual species is quite old. The Gomphidae are called the clubtails. That's probably sister to the Sasquatch food, to the petal tails. And there's around 1,000 species of these. We think that reproductive behavior may have driven this massive diversification in this group. The Cordula is the next group in our phylogeny. These are really striking. They're sometimes called spike tails because females have this behavior where she lays her eggs in muddy banks of streams. And it kind of looks like the darning needle is kind of spiking her eggs into the soil. And then we come to the final group, which is the group that first got me interested in studying dragonflies. This is what I worked on from my PhD thesis, the superfamily Libellulidae. So when I started working on my thesis, there was a lot of mystery about this superfamily. Initially, when people were first working on this in the 17 and 1800s, people thought maybe there was just one family. And then over time, in the 1920s or so, people expanded this to maybe being 3 families. And then others said, well, actually, maybe it's 5 families. And by the time I started my PhD, it expanded into having many, many families in this one kind of superfamily. Most of the work that was being done was based on wing venation. But it turns out that for dragonflies, wing venation is actually a really big part of their flight story. And many of the patterns that people were using to make their phylogenies, they actually were really correlated with flight, and they weren't good representations of evolutionary history. So when people started, like myself, when we started doing Sanger data sets, we were sequencing, mitochondrial nuclear genes, we were recovering phylogenies that looked very different from what the morphology phylogenies had looked like. And so there was a lot of debate about how they were related. And in general, much of what I had for my big magnum opus of my thesis was a polytomy, which means that we had really no resolution for this particular group. It's a lot of effort to come up with that. One thing I was able to find was that many of the subfamilies people had defined were not valid, and there was only one, the Libellulidae, that was recovered as a valid subfamily. So since 2008, we've kind of asked this question, would maybe having more data help us resolve this group? Or maybe having more taxa, could that help us resolve this group? Because I was just a humble graduate student. I couldn't do that many taxa. There's over 1,500 or so, 1,500 species, and I certainly didn't do that many. Well, one way to look for more taxa is to look at collections. And collections are really an invaluable resource. We have collections worldwide. The first collections, like people first started doing cabinets of curiosity, where you would have these little collections on your journeys that you would store. And eventually, the very wealthy people in Europe started actually in Austria, the first natural history museum. And these collections, many of them are quite old and have a really rich biodiversity. So for the American Museum of Natural History, we have over 300 objects in our collections, 30 million objects in our collection. Sorry, we have more than 300. We work hard. 30 million objects in our collection, and 23 million of them are invertebrates. And of those, around 65,000 are dragonflies and damselflies. So we can look to these collections, perhaps, to get more livability sequenced and answer some of these questions. But when we're using museum specimens, sometimes there's these other questions that come into play. So why is it that the American Museum has 30 million objects? Well, in part, it's because we went to a lot of places, we took a lot of things, and researchers in the countries from which we took the specimens often don't have access to those taxa anymore. So there's a lot in the museum world, there's a lot of discussion and debate about how we can make specimens accessible, who has access to specimens, and who should have access to specimens. But we also had this question of whether or not we could even use these specimens for DNA. The loftier social questions I can't answer yet, but I can tell you something about whether or not there's DNA in our collections. But just know, for those of you who are interested in talking about what we're doing on this forefront at our museum, these are the big questions that we spend a lot of our time debating. So in terms of the question of whether or not there's usable DNA, in these old specimens, our specimens are about 150 to 200 years old, or about our oldest dragonfly and damselfly specimens. So we wanted to ask this question, and we used specimens from the American Museum of Natural History and from the Naturalis Museum in Leiden in the Netherlands. And indeed, there's actually quite a lot of DNA in there. We thought maybe damselflies, which are tiny little things, would have less DNA, perhaps, that was preserved over long periods of time than dragonflies. We thought size would be a factor, but we actually don't really see that. For the purposes of making phylogeny, having 100 genes, you can make a pretty great phylogeny. So even for the tiny damselflies that had, not that much DNA recovered, they still were able to sequence quite a few loci. One thing that we did notice is that the American Museum specimens were kind of not great compared to the Naturalis specimens, and this probably has a lot to do with climate. So historically, all of our specimens are stored in curators' offices. And if you've ever been to New York in the summer, it is a hot, humid place. And so we think that that has something to do with the degradation of DNA, why we were able to recover fewer loci than the specimens from which we got from Holland. But in general, we were recovering, even for things 1909, 1911, we were recovering a lot of DNA, certainly enough DNA to make great phylogenies. Of course, you could say, what if you're just sequencing fungus and bacteria? Right? Maybe that's the DNA we're sequencing. Well, you can test that by, reconstructing phylogenies using the specimens that were very old and the younger specimens. And so the ones that I sequenced that were very old, that we sequenced, I have the year of collection indicated in red. And I'll just point to some here. 1937, 1931. My granddad was born on January 9th, 1921. So I made an effort of finding a dragonfly that was that old so I could sequence one from his birthday. And sure enough, we're finding that congeners are grouping with congeners. So things from the same genus are grouping with others from that genus, and same for conspecifics. So Somatochlora is grouping with Somatochlora, the Symphymis, the Chlorogompus, and so on. So we have all of these extra taxa that are now available to us. If we now know that we can use these museum specimens that are very old in this work, that allows us to ask this question of, can we add more taxa? So from when I was in graduate school, we sequenced maybe 8 genes, and it was a really big deal to do 8 genes. And now we're able to, with one of our probe sets, sequence 92 genes, and then the other probe set sequence 1,000 genes. And so we have this huge, massive data set. Probably going to get the answer, right? It's a polytope. That's what we found. We threw a bunch of money at it, a bunch of time. And what we found is that we still have no resolution for the livability. When we look, we can use something called bootstrapping, where a higher number indicates that we feel more confident about a relationship. A lower number means we feel less confident about a relationship. And we find that the numbers that for the backbone of this part of the phylogeny are pretty low. This could mean that it's what we call a hard polytomy. Maybe these dragonflies diversified really rapidly, and there just isn't a lot of signal, not enough signal for a computer program to really sort them apart. That could be real. That one subfamily that I have found with standard data as being strongly supported as monophyletic, the libellulini, we still recover that as monophyletic. So we've tried using transcriptomes. We've tried using 500 loci. We've tried using 1,000 loci. Did more data help? No. Did more text help? Well, not yet. I mean, we haven't sequenced all of the livability. It's possible that adding a few more genera that we haven't yet done is the one thing that you need to actually have everything fall in place and kind of answer this question. So we're kind of holding out some hope for the livability, but it's possible this is one of those questions that might just be really difficult to answer. We do feel confident now, at least we think we have sort of an understanding of how the true tree of life is for the Zygoptera, the damselflies, the Anisozygoptera, and the Anisoptera, the dragonflies. So once you have a phylogeny, then you can start doing the really cool stuff. That's when you roll your sleeves up and you can start answering questions. So one of the questions that I've always wanted to know is, how old are these animals? How we know that we have these carboniferous dragonflies from the Carboniferous. That's a lot older than what the fossil record suggests. And I'm particularly interested because I want to know about the evolution of certain flight behaviors. I want to know about the evolution of color. I want to know about the evolution of certain types of vision. So we can use molecular data and fossil data together, and that can allow us to do what we call divergence time estimation, estimate the age of particular nodes of our tree. And when we do this, we find that damselflies seem to have diverged around 225 million years ago. This is when the Earth would have looked something like this, with the continents arranged thusly. The cool dinosaurs like T-Rex, not even on the scene. This is what we're working with, that would have been kind of walking around at the time. Interestingly, for dragonflies, we find the same time of origin, around 225 million years ago. So now that we know something about the evolutionary history of dragonflies and damselflies, we kind of know how long they've been on the scene. We know that they've been probably shaping this very, very limited resource that we have on Earth, which is fresh water, upon which all life depends. They've been shaping it for over 200 million years. What did we do with fresh water? We dump a bunch of stuff in it, right? We're very good at polluting fresh water. And some dragonflies respond very poorly to the pollution that we put in the water. And as such, they can be great indicators of ecosystem health, especially for fresh water. The juveniles develop in freshwater. They go through a series of instars. Sometimes it takes 6 weeks. Other species, it takes up to 20 years. So things that happen in the watershed can have really long-term impacts on populations. Some colleagues from South Africa, John Samaika and Michael Samways, they actually came up with an index, the Dragonfly Biotic Index. This has been used widely in Africa and South America. We're using it in Ghana right now. And you can kind of look at which dragonflies are there, give them a score based on their pollution tolerance, and that can tell you something about how well the water quality is. And this is important because we want to ask questions about how these dragonflies and damselflies are responding to climate change. And when I say climate change, I mean habitat alteration, I mean aridification, nitrification, changes to water bodies and water sources. We know from the literature and from colleagues that dragonflies seem to be responding faster than any other organism on Earth. For example, there's a dragonfly called Anax, and Anax imperator used to only be found in Northern Africa. It, for the last 20 years, has had established populations in Sweden, because Sweden has warmed so much. So we're seeing kind of rapid change, and so some people call them climate canaries, because they're really kind of sentinels of what's happening. And in any case, when you have change, there's going to be winners and there's going to be losers. And the students and I were talking about this earlier, right before this seminar, about how sometimes the doom and gloom of climate change can really get you down. But there are winners, and sometimes it's important to kind of focus on the taxa that maybe are going to make it. And a good example of this is the blue dasher. This is what the blue dasher looks like. It's everywhere. If you've ever been to a pond, you've probably seen it. There are perchers. I've never really been super interested in the blue dasher because they're everywhere. I could always collect them. I assume they never left the pond from which they emerged. Not really an interesting story. Or are they? So I had when Ellie Gavitt, who's now my graduate student, when she was an undergraduate, she was doing her undergrad at BYU. She came to the American Museum for the summer. She needed a project, and she wanted to learn population genetics. We have so many blue dashers in the collection. Like, why don't you do something on the blue dashers? I'm sure it's not going to tell an interesting story because they probably never leave the pond from which they emerge. And what she discovered is that that is not the case. It turns out that they have a lot of genetic patterns that are shared among individuals that are spread out really, really far geographically. This is what's called a haplotype network, where each circle indicates a genetic pattern, and the size of the circle tells you something about the number of individuals that share that pattern. The tick marks on the lines indicate the number of nucleotide changes. So the first clue that maybe something interesting was happening was that there was a lot of sharing of genetic patterns, even for things that were from New Jersey, from Ontario, from Florida, from Tennessee. And she decided to do ecological niche mapping. This is where you take the distribution records that you have for a given species, and you can look at the environmental features of those locations, and you can predict what might be suitable habitat. And when she did this across time, she noticed in these heat maps, yellowish kind of green means highly suitable habitat, and dark blue means less suitable habitat. What she noticed is that there's kind of being an expansion westward. And what she noticed is that in some places in the West, she's from Las Vegas originally, there's actually suddenly a proliferation, a real range expansion in this taxa. And so in doing her work, she started trying to find out more about the larval behavior. And it turns out that these dragonflies can survive their entire life cycle in a swimming pool. So as humans have created water features in the desert, as we've created bird baths and swimming pools, they don't require a lot of water and they're not very picky. This is probably going to be a winner. So we may not have all the diversity of dragonflies there, but I bet you we're going to have blue dashers for a long time. The other dragonflies not doing so well. So the question of whether or not dragonflies are going to make it is something that kind of, it's near and dear to my heart. I love dragonflies and I want them to survive, but I also know they're really important in the ecosystem and in the food web. So we know that insects are declining. There's been lots that's been written about in the literature. People question whether or not we should use certain types of methods to really estimate abundance, or we should look at species loss. No matter how we've tried to quantify this, what we see is that across orders and across geography, insects are declining at a rate that is faster than we've seen before. A recent study that just came out last year on butterflies showed that for some taxa, like the cabbage white butterfly in North America alone, had decreased by 90%. So it's kind of a time that's a bit of a rallying cry for us to take some action. If you have data, which we do, we should really kind of work together to make some changes. But of course, the things that we have to do to make changes are going to vary depending on where you are. So I live in New Jersey, New York area. It's a kind of a concrete jungle, an urban wasteland, you might even say, although we do have Central Park, I guess. But the things that the insects are finding to be stressors there are very different than what you might be finding in Provo here. There in New York, it's light pollution. There's a lot of impervious surface. There's noise pollution. There's heat islands where it tends to be warmer in the wintertime, warmer in the summertime than it would be elsewhere. Reduced green spaces, lots of pesticide application, not done under the regulations that we see in agricultural systems, but you or I or my nana that just sees a bunch of pests and just sprays the heck out of it with another insecticide. This is really different from the types of stresses that we see in agricultural systems. In agricultural systems, sure, monoculture, that tends to support lower diversity, but that tends to be the way that we do our agricultural systems here in North America. Whether something is a staple crop or a specialty crop, they still often are treated with the same amounts of insecticides. I will say, though, that agricultural systems are incredibly regulated. There's more regulations than there are on agricultural systems than there are on my neighbor who wants to spray their lawn so they can have a perfect summer, right? Agricultural systems actually are fairly well regulated, but the insects that are in these fields are still suffering from noise pollution, invasive species, etc. Now, I do a lot of work in the Arctic as well as in the tropics. That was part of what this video that was playing out there was showing, our work in the Arctic. There's me trying to break open the ice to get some nymphs up in northern Finland. In the Arctic, when we go there, we're seeing dramatic changes. Every time we go year after year. And the amount of permafrost that we're seeing, the temperature of the water, the time when the first freeze sets in. These extreme weather fluctuations we're also seeing in South America. There are termite mounds that we've been monitoring since 2010. We go there and we check on them every year, make a little hole, a bunch of soldiers pour out. The last 2 times we went to Guyana, mounds were completely combusted because of increased forest fire. They had burnt to the ground. So we're losing taxa, even the ones that we may not know, like termites and cockroaches, they too are threatened with loss because of fire, extreme weather, temperature changes, and what have you. So it can be hard to know what to do. And at least where I'm living in the Northeast, one problem that we have is with trying to maintain a perfect summer. So I had someone come to my house last summer, and they had a little iPad with them, and they knocked on the door and they said, "Ma'am, would you like to enjoy summer again?" I said, "Yeah, I love summer. I would love to enjoy summer again." And on his iPad, he showed a bunch of insects. And he said, "We can get rid of all of these from your garden. You let us come and spray. It's like a mosquito control program, and we'll spray, and you'll be able to enjoy summer again." And this myth that your summer excludes insects, this myth that you can have a summer without flies and without other insects, that is what we're facing, especially in places like the Northeast. This idea that you're going to have a monoculture of grass, which is really great habitat for things like culex, which is a mosquito that likes to come out and start biting you right around the time when you turn the barbecue on, right around kind of dusk. That's when they come out and start biting. It's a real fight. Now, ideally, we would want people to turn their monoculture lawns into things that look like this. And I did try this. And just full disclosure to the group, I'll say this in front of the group. My homeowners association did not love my garden the first 10 years. It takes a long time to go from grass to looking like this. My garden now, finally, this is not my garden, but my garden looks something like this. Still too embarrassing to show a photo, but it takes a while to kind of go from a lawn into having a beautiful garden like this. And so people get a lot of pushback when they start to rewild their lawns. And so that is a real deterrent to getting user buy-in. As I said, we can make choices about the use of insecticides in our area. And I talk about this a lot with my town council. We have really beautiful green spaces in New Jersey, even though you would not know it from TV and movies. And a lot of that is maintained using insecticides, which is problematic. As I mentioned, I go to the Arctic a lot. And a big change that we've seen just even in the last 10 years is the amount of permafrost that's there. Permafrost has an active layer that kind of melts and freezes every year. And then this permanently frozen layer. And that frozen layer stores a ton of carbon. So as that melts, that actually releases carbon, which is a greenhouse gas, into the air, which further kind of exacerbates the problem of what we're seeing in the Arctic. We can make recommendations to people about energy use and carbon consumption. But I will say that for this kind of red part of the pie, my teenagers do not care. If I have the heat on and they're a little hot, they'll open the window, in their house, and they'll just leave it open. I will run it for a while. So I find it very hard to talk to people about energy use, especially when my teenagers would say, "It's not us, Mom. It's big corporations that need to change their energy use." And so getting people to move between their personal responsibility and the kind of more community responsibility, that is a challenge. So you could do these things. You could change your lawn. You could stop using insecticides. I could get my Nana to not have the guy come and spray her lawn. I could get my teenagers to close the window when I have the heat going. But all of those things are kind of small potatoes. I think the biggest thing that we could do really is just to vote for people. To vote for people who really care about green spaces. And like floods, vote for people who care about insects. Because if you have people who care about insects and they're going to advocate for insects, they are the decision makers that are actually going to make big change. Of course, we still need to have food security. And we're so fortunate. We're blessed with tons of food in our area. But there's lots of places in the world where we still need really integrated pest management so that we can preserve food quality and, of course, protect human health with things like malaria. But in general, insect decline is happening, and it's a global problem for which we need really global solutions. And what that means is that everybody has to participate. So these are my kids when they were younger. I used to take them to the field with me. There's Aishna holding a dragonfly like a boss, and there's my son in Guyana. There's no reason for anybody to be excluded. right? Science works better when we all work together. And if we have these really pressing problems that really affect all of humanity, we need everybody to be participating. So insects are essential. And I'll end by saying that museums are also essential because they're the things that kind of lock the history, and they're the baseline data to which all of our data are compared. We need more global solutions. And importantly, you should all stop whatever it is you're doing and become entomologists, right? We need more entomologists, we need more systematists, and you should join us. We have a couple of groups that are really actively working on the molecular phylogenetics and morphological phylogenetics of dragonflies, as well as looking at kind of bioinspiration in dragonfly morphological adaptations. So please join us or reach out to me if you have any questions or want to join. Thanks to my graduate students, and thanks again to Heath for inviting me.
Maybe we have just a little bit of time for questions, then we'll go eat some birthday cake. So any questions?
I don't remember the name of the group that you were talking about, which you were trying to resolve those roots. I was wondering if you've looked into introgression at all.
We have, and there does seem to be quite a lot of introgression happening along the backbone of that tree. And that may prevent us from really being able to tease apart what's happening. There's also this phenomenon called ghost introgression, where there's lineages that have already gone extinct that we can never sample that also introgress into the taxa that are living now. So the combination of introgression and ghost introgression definitely muddies the waters. This could be feel a little bit better though about the amount of time that we spent and the fact that we can't answer it. It's probably a real phenomenon, a real hard problem.
Any fun stories from your time in the field?
Oh yeah, sure. We go to the field a lot. And being in the field is a great way to kind of learn about biodiversity. I'm trying to think of a fun story. Well, the thing I'm most proud of, maybe in life, beyond having two great kids, I guess, is that one time I caught a helicopter damselfly with no net. So these are the really big damselflies. The wingspan is like this. They have long, slender abdomens. They actually feed on orb weaver spiders. They kind of hover in front of the spider web and pick the spiders off. The spiders drop to the ground when they fly by. They're kind of hard to catch because they have a fluttering mode that they fly in. It's very disorienting. So it makes it hard to know where they are. And I was in Guyana one time, and it was around time for lunch. And I was trying to find where this particular graduate student was. They kind of walked off. I said, I'll go to, and I'll look to see if I can find Dominic. I didn't bring my net with me. And we had been looking in earnest for this helicopter damselfly. And I saw one. And I'm like, what am I going to do? I have to try and catch it. So I stopped it for a while. I tried with my hand, but I managed to, with like my bandana and a bit of clothing, catch this damselfly just without a net. And that's maybe my sole accomplishment. I peaked, and it was all downhill from there. I went back and I threw it on the table, and everybody cheered. It was a very special moment.
When you talk about extracting DNA from the older samples, how did you go about doing that without damaging the specimen?
Well, we used a kind of modified extraction protocol so that we could still put the legs back with the original specimen. Normally, when you do an extraction, sometimes you do it over a day or 2 days. We actually, because it was older specimen, we figured the DNA would be degraded. We did an incubation step that lasted for an entire week. We added a few more of these extra chemicals that would prevent kind of breakdown of the DNA. And then when we were done, we took that leg that was intact and we put it back into the envelope so that it could stay with the specimen. Some people feel very passionately about their dragonflies looking perfect. People who collect dragonflies tend to like them because they're really beautiful. So when you smash them all up, they don't like that. So we always try and have them look as intact as possible and then put the legs back.
And where would you recommend kind of going to get more involved with entomology work?
There's a lot of great work in entomology. It's a really vast field, I would say. If you like systematics, there's so many groups for which nobody is working on them at all. And so there's lots of discovery to be made. If you like the idea of vector biology, there's a lot of people that work in vector biology, mosquito-borne illness, tick-borne illnesses. A lot of people work on food security and agricultural settings. That could be in specialty things like tree fruit, or it could be soybean or what have you. There's a lot of people who are really interested in forensic entomology because there are some things that like to eat dead things. And so you can use what insects colonize a cadaver to estimate the time of death. And so a lot of people do forensic entomology. Some people are really interested in like bioengineering, and like insects have done a lot of things first, structural colors that are kind of the bumps and ridges on beetle skin that makes them look kind of metallic. We now have fingernail polish that does that. That was inspired by things that we saw in the natural world. And there's house paint that has better ridges that are based in part by insect cuticle. The modern Velcro actually was modified slightly from the head arrestor system of dragonflies. So there's lots of people who are interested in bioinspiration, and they lean more into engineering and physics. So depending on the types of things that you're excited in, you can get lots of different types of like applications of entomology. And you can do that in industry. You can do that as a professor. You can do that as a curator, or you could do that in kind of startups. If you're making better fingernail polish colors based on beetle skin, people do that. It's actually a job. And if you want to get more involved, I would recommend reaching out to your local entomology group. There's a Dragonfly Society of America's local group in the Northwest area, the kind of West area, that you could join if you like dragonflies. The Entomological Society of America has a lot of great meetings. There's regional meetings that take place every year, as well as a national meeting. This year, the national meeting is in Ohio. And that's a great way to meet other entomologists, meet other undergraduates that are interested in entomology, or graduate students, and also to network. But you can also email me if you would like, and I can share more information. jware@amnh.org.
Just a quick thing, if you're interested, is a shameless plug. We have entomology this fall as a class you can sign up. So you can take a whole course with me, learning about all sorts of things on insects. So sign up for that. Yeah.
I am a certified pesticide applicator. Can you recommend any breeding for me?
Oh, yes, absolutely. Maybe I could exchange emails and I can send along some PDFs. We do have, did you get your certification through the Entomological Society of America? We do have a new certification program for pesticide applicators that has, I can forward that information along to you too. Pesticide application is like such an important role because you're actually interfacing with the people who are interacting with pests. And that is, I think, if we're going to change people's minds about insects, it's you, no pressure, right? But when people are only encountering insects as pests, they call you when they're in dire straits, right? They're often frantic and they need help for a particular situation that's happening in their home. You're the person that's able to talk to them about entomology and about the beauty that is all of the other types of cockroaches and termites. And I mean, nobody likes bedbugs, but there's lots of other hemiptera that are awesome. So, I mean, I bow my hat to you because I tip my hat to you because that's a really important role to meet with people when they're feeling the most panicked and get them to still maybe like insects and want to advocate for them.
What's your email again?
My email is my first initial J and my last name WARE, and then it's AMNH, like American Museum Natural History .org.
I think we're going to end there on that one. Let's give one more round of applause. We're going to head out to the atrium. We're going to sing happy birthday.